Go back to it. Pull up my chat. Ah, the joys of Zoom. I don't miss this. And I'm really not going to miss this once we can go back in person. All right. So this is my lab. I have a lot of undergrads that work in my lab too. So all the undergrads who are in my lab are paid. I don't have volunteers in my group. And we're a mix of PhD students in neuroscience, cognitive science, and probably data science once that PhD program launches. And then undergrads that are in cognitive behavioral neuroscience and psychology, cognitive science, computer science, math, data science, and so on. And so talk a little bit about the data science side and my background. If you've taken a class with me, you've heard this, so I'm not going to belabor the point, but if you haven't heard it, after I finished my PhD in neuroscience, I got recruited by a friend at Uber back when Uber was an 8-person startup called Uber Cab to help them build out their data science team. And so this is back in late 2010, early 2011. Data science wasn't really a thing yet. There was like 2 places that had data scientist as a job title, Facebook and LinkedIn. And I was asked to try and help. This is when Uber was still only in San Francisco, try and help figure out how to use the data that Uber was collecting about the position of the drivers and like where people got picked up and dropped off to try and figure out how to make the service better. So I hired their first data science team, worked on the data strategy, and then more importantly, met a bunch of really amazing people who were in the early days of data science. So that includes like UCSD alumnus Vijay Patel, who was the first chief data scientist for the United States that worked in the Obama White House. And I'm going to skip over all these kinds of things and just say that it was a pretty exciting time to work for a startup that became a worldwide phenomenon, and figuring out how to leverage those data to look at, for my interests, like how do people move around cities. And there's, I can come back to these, but I don't want to spend too much time on the actual like history side of things, because I do want to have that conversation with you all. And then on the neuroscience side, when I left Uber to go back to neuroscience, which is what my PhD was in, and what my lab focus is largely on, I'm very interested in how the 86 billion or so neurons in our brains actually communicate with one another, which puts me sort of in the camp of computational neuroscience, which is what are the computations that the brain performs that allow us to think and behave. And so in order to understand that, we not only leverage theory and computational modeling and simulation, we also actually do real recordings of neurons and neural activity during different kinds of behaviors. But in humans, we are limited by the kinds of recordings that we can do because we've got skulls that are in the way, and therefore I can't put electrodes into people's brains like people who work with animals do, except for very rare cases where people are already undergoing invasive brain surgery and already getting electrodes. These are like recording devices that can let us record brain activity implanted into their brains as part of their normal medical procedures. And so what we do is we work here at UCSD. It's with the neurosurgeon. Her name is Dr. Sharona Ben-Haim. She works with patients who have very severe epilepsy, which is a disorder that causes seizures. And for most people who have epilepsy, these seizures are relatively easily controlled by anti-epileptic drugs. But for whatever reason, in some small percentage of people that have very severe epilepsy, these anti-epileptic drugs don't abate the seizures. And so these people still have multiple consciousness losing seizure events per day. And obviously this is very debilitating to the quality of life. And so in those cases, these people often choose to undergo an elective brain surgery where the surgical team has to identify exactly what part of the brain is causing the seizure. The fancy term for that is the epileptogenic zone, or the zone of the genesis of epilepsy. And in order to identify that part of the brain, none of the non-invasive brain imaging methods that we have, like functional magnetic resonance imaging or EEG, are of sufficient resolution to allow us to identify exactly where that seizure is coming from, because the surgeon has to go in and actually remove a part of the brain. And so because of that, these people undergo several stages to their surgery. The first stage is a large part of the skull is surgically removed. Electrodes are implanted directly onto and into the brain for like 10 to 14 days. While the patient is hanging out in this epilepsy monitoring unit at the hospital with all these electrodes in their brain, just like watching TV, playing video games, reading, hanging out with their family who's visiting them, waiting to have enough seizures so that the team can say, aha, now we know exactly where the seizure is coming from. Then once they identify that region, they take out all the electrodes, remove the part of the brain that's causing the seizure, put the skull back in, sew everything up, and the patients are, if not effectively cured, their quality of life is tremendously improved. And the reason that this warning is here with this long preamble is I'm about to show you a video I took in the operating room of somebody undergoing this period of awake brain surgery. So this is right before they're about to remove the part of the brain causing the seizure. The warning is here is because I have had a student faint in class before because of the sight of blood. So you are warned. I'm going to give you a countdown to when you should stop looking if you are squeamish, and then I'll tell you when you can look back. So 3, 2, 1, look away. Here's the video.
Chair. Train.
Okay, and you can look back. Everybody good? All right, so there's only a couple of folks on here. So what do you all notice in that video? Anything in particular? Anything stand out?
Maybe this isn't what you were looking for, but it looked like the membrane with all the numbers on it was moving. It was like
kind of pulsing, right? Did you also notice the numbers? Anything else? Those are the big ones, right?
Was that the brain?
That's the human brain. That person's awake during that part of the brain surgery.
The brain looks different than I thought it would look.
So all of the brains that I have held have been sort of dense and kind of rough, like tough, thick. And that's because in order to extract the brain out of head post-mortem, the brains have to be chemically treated so that they're able to be removed, like touched. Because otherwise, in the head, in a living person, the brain has the consistency almost of jello. It's very, very soft. I mean, it's mostly fat and water. So it's not a lot to it, right? And so the brains are very delicate. And to address, I think it was Jan's point, even though the brain is only about 2 to 4% of the overall body mass, it actually gets about 20% of the cardiac output. So with every heartbeat, it gets about 20% of the overall blood flow, even though it's only about 2 to 4% of the mass of the body. So it gets a huge amount of blood for its size. And that's because brains are very, we say, metabolically expensive. It takes a lot of energy to keep brains running compared to other tissues in the body. Because of that, because it gets so much blood, with every heartbeat, it kind of like pulses and inflates a little bit. That's why the brain looked like it was pulsing, because with every heartbeat, it gets blown up like a balloon a little bit with blood, because so much blood goes rushing to the brain with every heartbeat. Those little numbers that you saw on the surface of the brain are literally pieces of plastic with numbers on them. So the surgical team is quite large. There's the main neurosurgeon, who is the sort of like the final point of the contact. That's the person. Dr. Benheim, Sharona here, is the one that's doing that surgery. And she has a surgical team of assistants in the operating room with her, unsurprisingly. And in this case, this patient, this was a patient not here in San Diego, actually, this was about a decade ago, had their epileptogenic zone, the part of the brain causing the seizure, close to a part of the brain that gives rise to like speech or language or motor control, so body movement. Because of that, they want to make sure, because everybody's brain is a little bit different, when they go in to remove the part of the brain that causes a seizure, you could imagine if the brain is, like a ball here and the seizure point is here, they could go in and remove it this way, or they can go in and remove it that way, or they can go in this way. And so they want to make sure that the path that they take to get the part of the brain that's causing the seizure doesn't go through a speech or language or movement part of the brain, because the last thing they want to do is, when they're doing the surgery to move the part of the brain causing the seizure, also damage tissue related to our ability to speak or understand language or move. You don't want to cause the patient to wake up partially paralyzed. And so because of that, the patient during this procedure is actually woken up for a little bit during the operation. And the surgical team stands there with a little electrical stimulator, and they electrically stimulate different parts of the brain to see if the stimulation causes like movement of the fingers or tongue or face muscles. And then they have the person read or listen to a story. And so you can imagine if the person is sitting there reading, Jack and Jill went up the hill to, and right when they're about to say fetch, the surgeon stimulates a part of the brain, and the person goes, fetch a pail of water. Then what they do is they take a little piece of plastic with a number on it, put it right on the place of the brain where they stimulated, and they'll write down on an iPad, stimulation site number 47 causes speech arrest. And that tells them to avoid that part of the brain when they're going in to remove the part of the brain causing the epilepsy. So neuroscience is wild. During the 2 weeks or so that the patients have the electrodes permanently implanted, so before this last stage of the surgery, when they've got the electrodes in their brains, we approach them in our lab and we say, we ask if they would be willing to do our memory or attention kinds of studies, because we can record human brain data from awake people that are doing very complex cognitive tasks in a manner that is otherwise impossible to do. Normally, you can only do invasive brain recordings from animals. And even if you do recordings in animals, they can't understand complex human cognition. So this is an unparalleled opportunity to understand how the brain might give rise to complex cognitive traits in a way that you can't do otherwise, non-invasively, because you have direct access to human brain activity, even down to the level of like being able to record from individual neurons. So this slide here is just to show it's a panel from one of the papers that I published a couple of years ago. You don't have to understand it. It's not really critical. It's just saying we can use these invasive brain recordings to look at potential mechanisms by which different brain regions communicate with one another. And in particular, one thing that my lab is focused on is this idea of neural oscillations, which is if you stick an electrode into anything's brain, sometimes you'll see these rhythmic, regular fluctuations in the voltage of brain activity. And we call those oscillations. They're brain rhythms, they're rhythmic, and they're the first signal ever recorded from a human brain back in 1929, when the first EEG was recorded. It's a pretty obvious signal in certain parts of the brain. We still, almost 100 years later, don't really know what they do, or if they do anything, or if they're just like a weird biological byproduct. And so that's the kind of data we collect. And one of the things my lab is interested in, I'm not going to go over the cortical organoids too much. This is something that we do with a collaborator. I'm not a cell biologist, so I'm just going to wave my hands and say, through the magic of cell biology, it turns out that you can take a skin sample from somebody, like a cheek swab, trick those skin cells into becoming stem cells, and then you can grow little tiny human brains in a dish. And then we could do experiments on these little tiny human brains in a dish. These are only about 100,000 neurons, about 4 millimeters in diameter, about the size of a bee's brain. Just to remind you, human brains have 86 billion or so neurons. These have 100,000. They're tiny, but it allows us to study human brain activity and development in a way that you otherwise cannot. Also, it would be impossible to do. So the data science stuff, my lab is fixated on writing a really good code that properly turns our data that we collect into numbers that we can interpret. And I know that sounds absolutely ridiculous, but to me, that's one of the like cores of data science, is how do we turn the data that we see into numbers that we can later analyze, and those numbers be interpretable, right? And so the example I like to give in my data science classes is like, so there's a question: how can you resolve neural circuit activity from those oscillations? That's a big question. I'll get to that. Let me finish this thought. So when we talk about you know what? I'll actually go all the way back to the Uber example, right? So when I was working at Uber, one of the things that we were curious about is when you open up a phone for any ride sharing service, right? It'll say like the nearest driver is 5 minutes away, 6 minutes away, 7 minutes away, whatever. And so one of the things that I had done was look at what is the probability of somebody who opens up the phone to look at the Uber app of then once they see how far away the nearest driver is, of them actually going and requesting a car and completing that trip versus shutting the phone off and saying, no, I don't want to deal with it. And so this blue line here, I should say blue curve, is showing the histogram or the relative number of rides completed on the y-axis as a function of, on the x-axis, the ETA, the estimated time of arrival, excuse me, of the nearest car. And so what you can see is the peak is somewhere between like 2 and 3 minutes. People are most likely to take a trip between, if the nearest driver is like 2 to 4 minutes, let's say. If it's actually only 1 minute, people are less likely to complete the trip. We sort of joke that this is like the too convenient. You're like, you know, still in class or at a restaurant and you like haven't paid your bill at the restaurant or haven't packed your stuff up in class or at your office yet. You still have to go to the bathroom or something. And so you're like, the driver's too close. Like, I don't want to request it now because otherwise I'm going to be rushed or like they're going to have to wait for me and I'll feel weird, right? So you close the app and you'll do it later. And so you have this dip here. It peaks between 2 and 4 minutes and then it decays exponentially. So the peak is at 2 to 4 minutes and then the probability of completing a trip exponentially decays as a function of time. And so we said, well, what if we find where that peak is and then just fit an exponential decay function, figure out what is the half-life essentially of this curve. And this curve tells us roughly how patient a given neighborhood is. So let's say you do one of these curves for every arbitrary neighborhood in a city. And one neighborhood looks like it does in the blue curve here, where it peaks at 2 to 4 minutes and then very rapidly exponentially decays. Another neighborhood that's like maybe way out far from the center of things, people are willing to wait a lot longer. And so that curve doesn't slow down. Imagine the gray here is not actually it, but imagine it looks more like the gray, right? It decays quickly, but not as fast as the blue one. And so you can parameterize these curves by fitting mathematical functions to them. And those functions can be interpreted as, say, this decay rate here as a function. You could interpret that potentially as like the patience of a neighborhood. The more quickly that curve decays, the less patient people in that neighborhood are for that service. You might say, oh, the nearest driver is 12 minutes away. Forget that. I'm going to open up Lyft or, you know, I'm going to grab a bird scooter now, or I'm going to walk. Right? Imagine that. That's an old guy thing to say. But anyway, so you could figure out ways of fitting equations to these curves and turning them into numbers that are interpretable. And so this is something that I spent a lot of time doing at Uber. And the argument here is that with proper parameterization, which is how do you turn your data into numbers that you can interpret, you can run large scale analyses that are otherwise not possible without doing that kind of parameterization, which then gets us into sort of the neural data science side of things, which I'm going to skip over, but we've got a lot of software that my lab has written in Python and stuff for you to analyze data. But I'll go back and address some of the questions really quick. Can you resolve neural circuit activity from neural oscillations, or can you determine neural cell types with neural oscillations? So I have to be very clear here. When you take a intro to neuroscience class, I mean, really even through grad school, take a neuroscience class, you learn that neurons, what does a neuron do? Anybody want to take a stab at trying to answer that question? You're in cogs, you should be able to do it, right?
The question was, what's a neuron?
What does a neuron do? What does a neuron do?
It carries messages in the brain.
It conducts a signal, carries messages in a brain. How does it? It receives and aggregates and sends electrical signals. And then how does it send those electrical signals? What kinds of signals are they? Do you remember?
Electrical and chemical ones.
Mm-hmm. Well, they're electrical ultimately, right? They use chemical messengers that open ion channels, and then the neuron membrane changes, its potential changes. You get a potential gradient. And once that potential crosses a threshold, they do what? What's the name of the signal? There's like a special name. Anybody remember?
Action potential.
Action potential. Why is it called an action potential?
Because it resets the voltage of the cell.
Right. And so it's a sort of like, like binary kind of signal. And so what happens is you've got the dendrites, the branches of the neuron, and those receive the inputs across the synapse, right? And those inputs are electrochemical chemical messengers, right? Neuromodulators, neurotransmitters, right? And those bind to the postsynaptic neuron, those dendrites, which opens up ion channels or closes ion channels, depending on what it is. And that changes the potential of the cell membrane, which then builds up until it hits a threshold. And then it sends an action potential down the axon, which then causes the release of neurotransmitter at the end of that axon into the synapse, right? That's the standard model of what a neuron does. It turns out neurons are way more complicated than that. They're not all or nothing binary signaling units. There's many classes of neurons that are graded action potentials. That means that they're analog. They actually share ion channels with neurons. So you have like a single ion channel that bridges between two neurons. So they share their membrane potential roughly. Action potentials can go retrograde. They can start at the axon and go back into the soma, the cell body of the neuron. We don't know why. We don't know what that does. Anyway, but when you record an action potential from a neuron, that's like the gold standard of how we understand how neurons work, right? You record and you say, I presented this animal a stimulus, I'm recording neuron activity, and I see that it sends action potentials, and those action potentials increase their rate. So there's more of them when I flash a light at this animal and I record from its visual cortex, right? But you're recording from those neurons, when you record an action potential, you're recording the outputs. This is the signal that is leaving that region of the brain. So you stick an electrode in, you record action potentials, that's the signal that's leaving. When you record oscillations, or what is called more broadly the local field potential, so it's a slightly bigger electrode, and you're picking up this like big aggregate kind of signal. That's not the action potentials of the neurons that are in that region sending their outputs. What you're largely picking up are those postsynaptic currents. So when the ion channels open, you get that you're inducing a current, which is changing the charge across the membrane. Right? And those currents are mixed between excitatory, modulatory, and inhibitory currents. And there's tens of thousands, if not millions or tens of millions, depending on your electrode size and impedance, of these currents that are coming in that you're picking up. So the action potentials that you're picking up are the outputs, and the local field potential that you record in these invasive recordings in humans or animals or at scalp EEG are largely the postsynaptic potentials coming into the region. And so sometimes they oscillate, sometimes they don't. But it's really important to say, what is it that you're picking up? And believe it or not, the reason I bring this up is because most people doing like neuroscience, not really super duper clear on this difference. And so it's important to know that action potentials output EEG basically is the incoming inputs from all over the brain, right? So most inputs are local. So neurons that are in a region sort of tend to communicate with each other locally. But a lot of the inputs are coming from all over the place. And so it's all mixed up. But my lab, like I said, we write code to try and understand what these signals that we're recording are and how we can properly measure them. And so like here in A is an example of a very clear oscillation in the hippocampus of a rodent running a maze. And this is called a theta oscillation. You could see there's approximately 1, 2, 3, 4, 5, 6, 7, 8 cycles per second or so, which means it's an 8 hertz oscillation, which is pretty standard for the hippocampus in a mouse or a rat running a maze. Most data doesn't look like this. You don't see really clear oscillations, but if you record from a mouse or a rat running a maze, record from the hippocampus, you see a very clear, strong 6 to 12 hertz oscillation that's called the theta rhythm. And so normally what you would do is you would write code to filter your data to make it clean, so you get rid of these jagged little spikes, and then you would analyze, the amplitude of each of these little peaks or something like that. That's how people traditionally analyze it. And what my lab is super interested in is taking advantage of the rich information in the data that we record. And like I said, writing a lot of code to then analyze it. And so what we do is, this is an example of what's called the bicycle algorithm, which you can install for Python. And that comes from the cycle by cycle analysis of neural oscillations. So it's an algorithm that takes your raw data, tries to find if there's an oscillation in it. And if it finds an oscillation, it then finds every time it crosses 0 volts. So here it says, aha, it crossed 0, 0, 0, 0, 0. This then allows you to break up an oscillation into the individual cycles. And then it then finds in between each one of those 0 crossings the maximum or minimum. So it finds the peak or the trough. And then it sort of iterates through, and it allows you to then take each individual cycle of your oscillation. Now this is 8 times a second. Sometimes they're as slow as once every 10 seconds, and they can be as fast as like 60 to 100 times per second. This is an 8 hertz oscillation. It then says, how long does it spend in the peak? How long does it spend around the trough? How long does it take to go from the peak to the trough and then the trough back to the peak? What's the period of each oscillation? What's the amplitude? How high is the peak? How deep is the trough? And so on. And so every single oscillatory cycle we can break up into all of these interesting parameters. And now we've taken what is usually a filter the data and then say how on average across this 1 second, what's the amplitude of this theta rhythm. We now can take our data to each individual cycle and say, give us all of these parameters and see if any of them are physiologically meaningful. And we can do that either from a pure data mining standpoint, just find correlations and structure in the data in a totally blind kind of manner. So that's an unsupervised learning approach, right, and machine learning. Or we can flip it and say, given the physiology of what we know that generates these oscillations, what parameters do we think are going to be important if we want to try and infer, going back to Jan's original question, the local neural circuit activity from those oscillations? So we take a 2-pronged approach. One is an unsupervised machine learning approach, which writes really tested code to turn these data into parameters, numbers, and then find structure just in the data, unsupervised. And the other side is run simulations about what we think are the generators, the neural circuit generators for these different signals are, and try and do a bottom-up approach, which is, given the constraints of biology, what features do we think biologically would have to change to give rise to different shapes of these rhythms? Does that make sense? And this is the approach I took at Uber too. We would do this sort of top-down approach of, turn our data into numbers and then mine through it for patterns, and then also run simulations of cities using historical data. And so the last project I had done for Uber was, got picked up by the press briefly as like simulating an AI simulation of San Francisco, which is using historical data from Uber of like every trip that had occurred. You can say, well, we know statistically at any given hour or minute of any given day of the week, probabilistically, where are drivers who work for drive for Uber most likely to be in a city, and when do people happen to appear statistically requesting rides, right? Like the nadir is at like 4 a.m. You get very few people requesting rides, and the peak will be Friday, Saturday nights at 10 p.m. to 2 a.m. And so you can do Monte Carlo simulations of using historical data to try and simulate forward different parameters, different simulations. You can run a city simulation over and over and over again, tweaking certain things in your like dispatch algorithm to determine like which driver gets paired to which client, to figure out which algorithm might perform best in simulation. And so this is something my lab is really big on, is sort of this 2-pronged approach. One, unsupervised data mining kind of approach, and then the other one more of like taking the biological reality of what we know generates these data and simulating it to try and figure out how to pair the data that we have with biology. And then you can do a bunch of cool shit after that. But it's now 40 minutes. And so I really do want to stop now and let you all chat.
For your simulation, what kind of models do you use?
For simulation in neuroscience,
Yeah.
nothing sophisticated. We don't do like really crazy, like sophisticated simulation stuff. What we often will do is, it depends on what we want to do, to be honest. So first pass, what we almost always do is what's called an Lnp model, which is linear nonlinear Poisson. It's a ridiculously complex name. I feel like all machine learning has like overly convoluted and complex sounding names for things that end up being pretty simple. So what you do is you can simulate action potentials in time using a Poisson distribution. And neuron spike rates are approximately Poisson distributed. They're not random noise, white noise distributed, Gaussian distributed, right? They have a Poisson distribution or close to one. And so you can simulate thousands of individual neurons with these Poisson distributed spike rates. And then you do what's called a convolution, which is we know what the postsynaptic excitatory and inhibitory and modulatory currents that give rise to local field potentials look like. And so we take those spikes, take your postsynaptic kernel. Actually, let's see if I have an example here. Oh, no, I don't have an example in this slide deck. But anyway, it's a double exponential kernel. And then you can convolve those with the individual spike trains, integrate everything together, and that gives you a simulation of local field potential. And you can change the amount of how much excitatory current or inhibitory current is contributing to your signal to see if you can infer, do the reverse inference of, given a local field potential signal, can you infer the relative balance of excitation and inhibition from that signal? Or given, let's say you, instead of doing Poisson distributed random spiking, you have certain groups of neurons that spike in bursts. And they have like a tight temporal clustering that comes in different rhythmic frequencies. Can you infer the temporal variance of the inputs, excitatory inputs that a region is getting based upon the shape of the oscillation that we see in the local field potential, for example? So that's often the first step, is doing these what are called Lnp models of the local field potential. If we want to get more complex and we want to check a little bit more sophisticated things, there's a package in Python called Brian, which is just a misspelling of brain, intentional. Brian 2 is the latest version, and that allows you to do a little bit more sophisticated modeling. You can even use like, you can model instead of spike times, which is what the Lnp model does, which is just, is a neuron spiking, yes or no, 0 or 1. You could do like Hodgkin and Huxley style modeling. Brian, sorry, Brian. Hodgkin and Huxley modeling of like individual action potentials, changing different parameters of like the calcium, concentration around the neuron, or the potassium or sodium currents that drive the action potential, and so on. But usually we don't have to go into that kind of depth. Although lately we have a new project that we might need to end up doing that, because my lab is getting very interested in the shape of individual action potentials. Action potentials, again, like I said, aren't all or nothing. They're very quick. They're over in about a millisecond and a half or so. But there's a lot of interesting structure in the shape of an action potential. And we're now looking to see if we can parameterize individual action potentials and like make inferences about the like relative sodium and potassium and calcium currents that are driving individual action potentials based upon the shape of the action potential. But anyway, we really like turning data into numbers and then playing with those numbers. That's really what my lab does. That's the essence of what we do.
For the unsupervised machine learning part, which algorithms do you use?
Again, really depends. The most common and simplest would be, if you want to try and see if there's any kind of latent structure to your data, just do like simple principal component analysis, PCA. But trying to think. Now, PCA with clustering tends to be the most common. We don't usually worry about using the most like sophisticated or complex. We don't do any deep learning, for example. We try and avoid black boxy kinds of models. We like to do things that can find structure in our data, like PCA, that you can then potentially interpret those components. So, yeah. So, do people have questions about, jobs in CogSci, or like how to go into grad school, or how to get like a research position in a lab? Like, what do people want to do? What are your concerns? I know you're all stressed out. Come on, it's like, what is it, week 7 of the last quarter of the pandemic school? You got to be pretty stressed out. What can I help you with?
Do you have any advice for getting jobs?
Yeah, actually. Make sure you have a good resume. Here, I'm going to share something. We actually just did, I don't know if she posted it yet. Let me see if I could find our resume workshop. Okay, I'm not going to sit here and Google for you all. I'll try and find it in a second. But anyway, over at the HDSI, the Hui Jielu Data Science Institute, Margaret Zulke is their Career and Alumni Services Advisor. And she and I got very frustrated by the resumes that we were seeing students put out there. And so we created an entire resume workshop in like a little half-hour long video about how to do proper resumes. She sent me the link. Here we go. I'll put it in the chat. So my number one piece of advice, and I tell this story in my intro to data science class, so I apologize if you've taken my classes and heard this before, but it's important, so I reiterate it all the damn time, which is a very common thing to see is if I'm at a university recruiting fair and I've got my recruiting booth and I'm trying to recruit people, people come up and they'll hand me their resume and it'll list like the classes that they've taken. And that's very impressive. You've taken a lot of machine learning courses. But so has everybody else. And you're talking about a large university that has, UCSD has a great reputation in the technical side of things, computational neuroscience, computer science, data science, math. We have a very good reputation in industry for these things. But we also have thousands of students in those domains. And you see all these resumes and they're like, everybody's kind of looking the same, right? And they'll list things like which classes they've taken. And I don't know what COGS 108 is. And so I look at a resume and it says COGS 108 introduction or, data science and practice. That doesn't tell me what you learned in that class. I don't know. It just has a course title. Is it in Python? Is it in Java? Is it in R? Is it in Julia? Is it in, I don't know, right? I don't even know what language it's in. And so those start to blur together. And so when you're looking for a job, you want to give yourself every little tiny advantage that you can, right? And so you want to make you stand out a little bit on your resume and your skills. So highlight the skills that you have learned. Don't tell me the names of your courses. Tell me the broad, big picture ideas that you have learned, right? Don't tell me that you've taken neural signal processing, COGS 118C. Tell me that you understand complex time series analysis and the Fourier transform. And, I don't know what else you might learn in that class, right? Because I haven't taught it. Or, you've taken COGS 108. You can say, look, I understand, the workflow of using GitHub and Jupyter Notebooks for data wrangling and visualization and report writing or something. I don't know how you'd phrase that for a resume. But the goal is listing things that you have done, the projects with like links to online resources like your GitHub projects links, or if you have a personal website, link directly to projects that you have done in your courses in that PDF. And so I look at that and I say, oh, this person did a project. One of the ones we did in this resume workshop that we ran last week was analyzing all the scripts of every episode of The Office to look at how different characters interact, and doing sentiment analysis of the text from the script to try and figure out what the relationship between different characters is and how it evolves over the different seasons, right? You can write about that in like 3 bullet points in your resume that'll take up just 3 lines, but it'll teach me that you understand natural language processing, that you understand sentiment analysis, that you understand how to come up with a clever and creative idea and then execute on that. And that then makes you stand out. So when we were at these recruiting fairs, and I would see all these resumes with people that have taken all these classes and they're listing all of the machine learning stuff, classes they've taken, I'm like, oh, okay, all right. And all of those resumes get put into a box, and we tape that box up and ship it back to human resources. And our hiring team would go through all of those resumes and try and find people to call in for interviews. But a handful of the resumes, instead of going into that box, would go into my backpack, and I would be on the phone on the cab ride back to the airport with a hiring manager directly saying, you need to bring Christina in for an interview right now before Facebook, Amazon, whatever, Netflix steals her away. And so your job as an undergrad is to maximize the probability that your resume gets put into the hiring person's or recruiter's bag instead of the box. And the only way to do that is by showing who you are, by like making your resume stand out a little bit more. And to do that, you have to do projects, or it helps a lot. You don't have to do projects, but it helps tremendously if you do these kinds of data-driven projects. And those projects can be on the data science side, like I said, analyzing sentiment analysis of stuff like that, or it could be you created a spiking neuron simulator on a website, or you did a cool visualization of how the Fourier transform works. Brian says, I like to read or go hiking instead of programming in my free time. How can I make myself and my resume still look good when I don't do lots of side projects? It's honestly tough. I play video games in my free time, and I can't put that on my resume, right? I'm still grinding through God of War right now, and like, I don't know, I can't say anything about that. And like Minecraft with my kids, right? The hard part is, your goal then would be, if you like to read or go hiking, figure out a way to turn your hobbies, the things that you like, into a side project. You can imagine, there's like different forums for hiking online, and it's like Yelp for hikes, right? Where people will post like star ratings for different trails and stuff in different cities. And you can imagine, writing code to scrape those and try and find, which cities have the best routes, or like text mine the comments that people leave on different hikes. Trails, to try and figure out, which ones are more difficult or more fun or have the best scenery, right? You can absolutely figure out ways of turning your hobbies also into projects. And so that way, at least they're kind of fun and interesting rather than super incredibly mind-numbing. But that's the reality of the job market. It doesn't mean you have to do that, right? You don't have to have all of these things in your resume. You will still get calls for interviews, even if you don't. What I'm talking about is ways of like really maximizing the probability that you're going to get the job that you really want. And it's all about sort of these numbers games. Yeah, exactly. Yeah, Christina, that's exactly what I'm talking about. Stuff like that would be fascinating, right? I would, if you made that and you created a like website that did that, I would go to that website. I think that would be super cool. Are people anybody thinking about grad school?
Maybe in the future, the far future.
I think grad school is awesome. I highly recommend grad school. And I'm gonna preface that by saying, I think most people feel like grad school is too expensive for them. And that is true if you go to like business school, law school, med school, or a master's program. But PhD programs in the sciences pay you to go to them. Exactly. Yeah. And so you're not going to get rich off of doing a PhD, but you're not going into debt. So, if you do a PhD in the neurosciences at UCSD, I think it's $34,000 a year stipend, and you don't pay any tuition. So the program pays your tuition. Actually, I do. The professors do. Whoever your professor is that you're working for in your PhD, they pay your tuition for you using grant money, and they pay you a stipend. So you're not getting rich. You're not making as much out of undergrad directly as your friends that go on to do software development jobs or anything like that. But PhD for me it's hard. I'm not gonna like sugarcoat it, but it's super fun. You get to spend 5 or 6 years really digging deep into something that you love. And I don't recommend you do it if you don't love it. So if you're like, neuroscience, cog sci is kind of fun, and maybe don't do it because you're sacrificing a lot of time and a lot of money. You're not paying for it, but you're sacrificing money because you're not getting paid as much as you could if you went for an industry job instead of a PhD, right? But it's enough to live on, and it was really fun. I very much enjoyed it, and it gives you, having a PhD opens up quite a number of opportunities also. Yes, Brian, that's also true. If you work for a company, there are quite a number of companies that will pay for your master's work for you while you're working at the company. That's absolutely true. And so that's one way of getting a master's without going into a lot of debt. Oh yeah, in a PhD program in the United States, you don't need a master's for. You apply directly into a PhD program, and you sort of get a master's along the way. So it's a little bit different in other countries though. You're interested in topics in bioinformatics, neuroscience, and data science. I've been trying to work in a few different labs or companies over the next years. Is that a good strategy for someone interested in grad school eventually, or should I just cut to the chase and shoot straight for grad school? I took 2 years off. So my undergraduate GPA was not good. And so I knew I couldn't get into grad school without a gap year or two. But I ended up getting very lucky. I mean, my whole career trajectory is based off of huge amounts of luck, honestly. And when I finished my undergrad in psychology, one summer before I graduated, I worked in a board and care home for people with like bipolar disorder and schizophrenia. So I had a little bit of clinical experience. And then I worked as an undergraduate in the lab, neuroscience research lab. And that was through a work study program to help pay off my student loans while I was doing my undergraduate work. And then those experiences helped me find a job at UCLA after I finished my undergraduate. I got a job at UCLA working in a neuroscience research lab full-time as like a staff researcher for the lab. And that then let me build up some research experience to become a co-author on some papers that then were enough of a compensation for my bad GPA as an undergraduate to let me get into grad school. So I think if you can manage to find a full-time job out of undergrad in a research lab, I mean, and if you go to like jobs.ucsd.edu, every university has a job site like this. And I took advantage of that after I finished my undergraduate. I went to all of these different university job sites and like put in keywords for, neuroscience basically, and applied to every single one that I looked like I had qualifications for. And again, you're not going to get rich as a lab tech, but it's a full-time job and it'll pay enough for you to be able to eat and pay rent.
So, based on that, like, if you think you want to do to grad school, but you don't know what you want to research, because like you have to apply to like the specific like lab, right? So you think it's better to like get a job and try to find what specifically you want to do?
You don't apply to a specific lab usually. So like the neurosciences grad program and cog sci PhD program here, both data science PhD program here is going to do the same thing, which is you apply to the program and an admissions committee looks over your application and decides whether or not you're qualified for the PhD program enough to invite you for an interview. Then when you interview, you'll interview with a bunch of different professors and try and see if your interests align. And then based on that, the program will accept you or not. So you don't really, strictly speaking, especially in the neurosciences program, you absolutely don't apply to a professor. Cog sci individual professors have like a little bit of say. So like if you're a new professor who doesn't have any PhD students, you know, you could say, no, I really want to bring in this PhD student, please, because they're super qualified. They might get like a little bit of a bump, but otherwise it's all up to the admissions committee. I mean, my PhD application, for example, when I applied for grad school, my application, and I did not get into UCSD cogs for my PhD program. I didn't even get an interview here, which I think is pretty funny. My application was to use fMRI to examine whether video game addiction is similar neural circuitry as drug addiction. Because the lab I was working for at UCLA at the time was a drug addiction research lab. So we studied people who were methamphetamine addicts, and then we scanned them as they were like held in the hospital, doing part of a rehab for a month as they're coming down off of their addiction to look at, you know, neural changes. And so I was like, well, I should take advantage of the neural addiction stuff. But, you know, what other things I'm interested in technology, and, you know, like nobody studies video game addiction. That's not really a thing. So maybe I do nothing related to that. During my PhD, I did nothing at all related to that, but that's what I wrote in my PhD application. So what you ultimately do in your PhD doesn't even have to look anything like what you actually wrote in your PhD application. But you should try and do some research as an undergrad. If you're considering a PhD at all, you should do so. You should try and find research experience in a lab either during your undergraduate or, you know, full-time work for a year or two after your undergraduate, just to see if it's something you enjoy. Because if you don't like it, again, it's going to be just a lot harder for you. And if you don't like it, then why are you doing it? Go find a job that you think is more fun and that pays better.
There's a question in the chat saying, like, if you the company sponsored programs, do you still work the job, or does the master's program become the job?
I didn't see that. The company sponsored. No, I think it's usually a nighttime thing. So I think you still have a full-time job, and then you have to do the master's on nights and weekends, I think. It may differ, but most that I've heard of are nights and weekends. So it's tough. It's also very hard.
I think it's like you also don't have to do like TAing and like stuff like that, right?
I'm not sure. We're getting into like very specific details of things that I actually don't know how that works for master's programs. Yeah, sorry.
So for the resumes, it's pretty much just making sure like your like how you describe the things you've done is like enticing looking enough. Or
well, and also the way that you describe them is in a manner that demonstrates the skills that you used, because you want to demonstrate skill mastery or proficiency, right? And so just listing classes that you've taken doesn't do that as well as saying, here is a project I have done that demonstrates me actually putting those skills to use. Does that make sense?
yeah, because I was thinking, because in my resume, like I've wrote down like the projects I've done, and every time I can get to an interview, like the interviewer's like, wow, your resume looks great, but then I also get like 70% like automatic rejections.
Oh, I think that's just normal. Unfortunately, you're going to get a lot of rejections. Rejections are just everywhere. That's kind of the standard. Unfortunately,
it's like I wasn't sure if like it was just everyone is good, so they're just going to reject a bunch of people, or if like there's something different like else that's like specifically wrong with my resume.
There is no magical thing that'll get you every interview. You know, you're going to get rejected a lot, and that's true when you're applying for jobs. It's true when you're applying to grad school. It's true when you're a professor trying to apply for grants. You know, it's you know, the grants that I write to keep my lab funded have a 10% funding rate. So 90% of grants that are written by professors that, you know, are in theory, you know, the ones that are leading the fields in this area, 90% of them are rejected.
Wow. I guess it's normal then, because I was thinking, because when I get an interview, they're like, wow, like you must have a lot of people asking to interview you. And in my head, I'm like, well, not really.
Well, I mean, the answer to that question is yes. In fact, I have a lot of interviews. What can you offer me? I don't know if you also saw the original Ghostbusters movie from like the 80s or whatever, but there's a line in there which is, if somebody asks if you're a god, you say yes. Right? If somebody offers you that kind of praise, you're like, absolutely, I am the best, and it's going to be very difficult for you to hire me.
I will say that next time.
Maybe don't say it exactly that way, but
I'll
find
say
out a
it a
good
little
way to
bit
say it
nicer.
that they, like you a lot. Okay, so Jan, how do you turn an idea into a research question and then generate an experimental design from that? How do you decide if that idea is a keeper? That's the magic of this kind of data sciencey side of things, right? Is talking with Brian about the hiking thing, right? You start with something that you like, and you say, okay, well, given this starting point of, I don't want to ask you what you're like, but like Brian also said reading, right? I love reading too. I read a lot of books, and I, particularly enjoy either biographies or sci-fi and fantasy. And so then you could say, well, maybe, I could look at different biographies written in different years and look at the different kinds of words that are used and say, like, are there gender differences in the words that are used in biographies? And has those gender differences changed over time? Using Google Books, which has like annotated, millions and millions of, or not annotated, optical character recognition, digitized millions of different books. Or you could say, have themes in science fiction changed over time? And so the trick is say, start with something you like, science fiction, and then say, well, I'm really curious, have themes in science fiction changed over time? What does that mean? That's very nebulous. Well, how do I quantify a theme? And then you could say, well, there's different kinds of science fiction, right? And like different books are classified differently, but like, how can I identify those? And I'll give you an example. Two friends of mine from grad school did, which is they took the text from the Hobbit books, the Tolkien books, and similar actually to that office thing I was telling you about. This is a pretty easy one. This is why people like doing it. Is they said, how often do different characters interact with each other, and how does the sentiment of their interactions change over the course of the book series? And so they said, well, we're going to define two characters as interacting if their names appear together in a sentence. So they find every period, and those are the stops. And then within each, between each period, they then said, okay, well, do multiple characters appear? If more than one character appears, who appears together and how frequently for any given chapter? And then what's the sentiment of each sentence, right? And so these are the kinds of things that you work through about. You take some big nebulous idea, you start it with a theme, something that you like, sports, movies, books, reading, hiking, right? Music. And then you say, well, what kinds of interesting things can I do given the kinds of data that might exist? And usually, transcriptions of like transcripts from different shows or episodes, lyrics for different sort of songs, the text of different books. Once you know what kind of raw data you have, you can then think about analysis that you can do on those data. And again, that's how do I turn this complex data into numbers that I can do statistics on in order for me to answer otherwise unanswerable questions? I don't know if that helps at all, if that was too nebulous. Another option is we actually have a data librarian on campus. Her name is Stephanie LeBeau. And you can email her and say, I'm very interested in doing a data-driven project. Do you have any resources? Because she can tell you what kinds of data are out there. She has access to huge amounts of data through the UC San Diego library network. And so she's usually pretty happy to talk with undergrads about project ideas and brainstorm with you. She's fantastic. So Google her name and email her if you're stuck on trying to find an idea.
What was that?
Hold on, everybody, 1 second. There you go. All right. Anyway, any other questions? Anything I can help with? Feel free to email me also, by the way, if you're like too shy to speak up right now.
Yeah, thank you for giving us all this advice.
Yeah, I hope it's useful. You know, I hope your quarters are going okay. I know this is not optimal, to say the least. And so, if I can help, please do let me know. Want to make sure that you all are, going out and succeeding and doing really well.
Just trying to get a job.
Yeah. Are you on the market right now?
Yeah, so I've been doing some interviews. I actually did an interview earlier today for like a machine learning engineer.
Oh, okay. If you want to email me your resume also, I have some like insider track knowledge about jobs that are not being publicly posted. And so I could try and, hit up some friends that are looking to hire people too.
Oh, that would be amazing. Thanks.
Yeah. All right. Good luck, everybody.
Okay, thank
See y'all
you.
later.
Bye.
Yeah. Bye.